Your education is your passport. So keep educating yourself. Key is important to open your life. Yes, we can. But how? You learn something every day if you pay attention. Push harder than yesterday if you want a different tomorrow. I'm Chitra Silvania welcoming you all with a happy heart. Today, we will start the second unit, which has three chapters: Principles of Inheritance and Variation, Molecular Genetics and Evolution. Principles of Inheritance and Variation explains multiple alleles, human blood groups, genetic control of RH factor, sex determination, sex-linked inheritance, karyotyping, pedigree analysis, Mendelian disorders, chromosomal abnormalities, etc. Today, we will discuss the first four topics. Genetics. What is genetics? Genetics is a branch of biology that deals with the study of heredity and variation. Do you know what is heredity? Inheritance of characteristics from parent to offerings is called heredity. And what is variation? Difference in characteristics among the individuals is called as variation. Who is called a father of genetics? Yes, it's Gregor John Mendel. He is considered as father of genetics. Genetics describes how the characteristics and features pass on from the parents to their offerings in each successive generation. The unit of heredity is known as the gene. A gene is the inherited factor that determines the biological character of an organism. Okay, let's move on to the lesson. First is the multiple alleles. The law of segregation in Mendelian inheritance reveals that all genes have two alternative forms. One is the dominant, another one is the recessive alleles. For example, you can take the tall plant and the dwarf plant, small capital T and small t. The tall plant is the normal allele or a wild allele, and the dwarf small t is the mutant allele. A gene can mutate several times, producing several alternative forms. So, what is multiple allelism? When three or more alleles of a gene that control a particular trait, here trait is character, occupy the same locus on the homologous chromosome of an organism. What is locus? Locus is a point, at a particular point or at the same point of the homologous chromosome. They are called as multiple alleles, and the inheritance is called as multiple allelism. Next, we move on to human blood groups. Multiple allelism occurs in humans, particularly in the inheritance of different types of blood groups. The blood group inheritance in human can be understood by learning about antigens and antibodies. You might have studied what is antigen and antibodies in your lower classes. So let us recall. What is antigen? Antigen is a foreign particle that enters into the body that is called as antigen. And antibody is a substance that is produced in our body to detoxify the antigen. Now, coronavirus is very, you may know about coronavirus, and it's caused by, it's a special type of antigen which causes a disease. So the composition of blood, different types of blood groups, blood antigen and antibodies were discussed in your 11th standard itself in chapter 7. Now we move on to ABO blood types. Multiple allele inheritance of ABO blood groups. Blood differs chemically from person to person, isn't it? So if you see the biochemistry of the ABO blood grouping system, where we will be discussing about the formation of different antigens on the surface of erythrocytes, that means RBC. The basis of these chemical differences is due to the presence of antigen, that is the surface antigen, on the membrane of RBC and epithelial cells. In human, the ABO system has established within the population. So there are A blood group, B blood group, AB blood group and O blood group. In case of A blood group, A antigen is present on the surface of RBC, and in case of B blood group, B antigen is present on the surface of RBC, and in case of AB blood group, both antigens are present on the surface of RBC. And in O blood group, the H antigen is present on the surface of the RBC. You heard that O individuals lack antigen in RBC. It's because O individuals have null alleles. Null means 0. And they do not go for any addition of molecules on the precursor molecule. Here the precursor molecule is nothing but the H antigen. It is already present in the form of H antigen, and this H antigen acts as an precursor molecule of both A antigen and B antigen. So, who find out this ABO blood group? Carl Landsteiner discovered two kinds of antigen called A antigen and B antigen on the surface of RBCs of human blood. You know the type O blood group, it is called as a universal donor. So, and the rarest blood group AB, it is called as the universal recipient, and it was discovered in 1902 by two of the Landsteiner students, von de Krastel and Sturley. Bernstein in 1925 discovered that inheritance of different blood groups in human beings is determined by a number of allelic series. The three autosomal alleles located on chromosome 9 are considered with the determination of blood group in any person. So, to determine the blood group in a person, the alleles are present in chromosome 9. The gene controlling the blood type has been labeled as L, capital letter L, after the name of the discoverer Landsteiner. Or we can call it as I, capital I, that is from isoagglutination. So what is agglutination? When two different blood groups are mixed, agglutination, that means clumping together of erythrocytes, that is RBC, occurs. So the I gene exists in three allelic forms, IA, IB and IO. IA specifies the A antigen, IB determines the B antigen, and IO allele specifies no antigen. Individuals who possess these antigens in their fluids, such as saliva, are called as secretors. Because saliva is secreted in our mouth, so it is called as secretors. Each allele, IA and IB, produces an enzyme, that is a transferase enzyme. IA allele produces N-acetyl galactose transferase and can add N-acetyl galactosamine, NAG. And IB allele encodes for the enzyme galactose transferase that adds galactose to the precursor, that is the H substance. In case of IO allele, no terminal transferase enzyme is produced, and therefore it is called as null allele, and hence cannot add NAG, that is N-acetyl galactosamine or galactose, to the precursor, that is the H substance. From the phenotypic combination, it is evident that the alleles IA and IB are dominant to IO, but co-dominant to each other. IA is equal to IB. I repeat, IA and IB are dominant to IO, but co-dominant to each other, that is IA is equal to IB. So the dominance hierarchy can be given as IA equal to IB, which is greater than IO. A child receives one of three alleles from each parent, giving rise to 6 possible genotypes. What is genotype? Genetical makeup. So genotype and 4 possible blood groups, that is phenotype. Morphological character. The genotypes are IAIA, IAIO, IBIB, IBIO, IAIB and IOIO. So we can see the genetic basis of ABO blood groups in this table. If the genotype is IAIA and IAIO, the blood type is A blood group and the antigen present is A. The antibody that is present in the plasma blood is anti-B. In the genotype IBIB and IBIO, the blood group type will be B and the antigen present will be B and the antibodies will be anti-A and anti-A. If the genotype is IA and IB, the blood group type is AB and the antigen present on the red blood cell is A and B. The antibodies may be neither antigen A or antigen B. In the genotype IOIO, the blood group be O and the antigen present will be neither A nor B and the antibodies present in the blood plasma will be anti-A and anti-B. Next we move on to Rh factor or rhesus factor. This Rh antigen is found on the surface of erythrocyte. It was discovered in 1940 by Karl Landsteiner and Alexander Wiener in the blood of rhesus monkey, Macaca rhesus, and later in human beings. The term Rh factor refers to immunogenic D antigen of the Rh blood group system. An individual having D antigen or Rhd positive and those without D antigen are Rhd negative. Rhesus factors in the blood is inherited as a dominant trait. Naturally occurring anti-D antibodies are absent in the plasma of any normal individual. That means the blood of an individual. However, if an Rh negative person is exposed to Rh positive blood cells for the first time, anti-D antibodies are formed in the blood of that individual. That means if Rh negative person is exposed to Rh positive blood cell, anti-D antibodies are produced. On the other hand, when an Rh positive person receives Rh negative blood, no side effects is seen. And these Rh factors are controlled by certain genes. It is explained by Fisher's and Ray's hypothesis and Wiener hypothesis. First we see Fisher and Ray's. Rh factor involves 3 different pairs of alleles located on 3 different closely linked loci, that is point on a chromosome pair. 3 different pairs of alleles in 3 different loci. This system is more commonly used today and uses the CDE nomenclature. Here the 3 different alleles are represented as capital C, small c, capital D, small d, capital E and small e in the chromosome pair 1. The possible genotypes will be 1 capital C or small c, 1 capital D or small d, 1 capital E or small e from each chromosome. For example, you can write capital D, capital C, capital E or small c, small d and small e. All genotypes carrying a dominant D allele will produce Rh positive phenotype. If capital D is present in that particular type of allele, it is Rh positive. And a double recessive genotype, small d will give rise to Rh negative. If capital D is present, it is positive. If small d is present, it is negative. Next we see Wiener's hypothesis. Wiener proposed the existence of 8 alleles. In Fisher's, it is 3 different alleles. Here in Wiener's, it is 8 alleles. R1, capital R2, capital R0 and capital RZ, and small r, small r1, small r11, small ry at a single Rh locus. They are present in a particular place. All genotypes carrying the dominant R allele, capital R1, R2, R0 and RZ will produce positive phenotype and double recessive genotype small r's will give rise to negative phenotype. If dominant character is capital R and small r is the negative character. Next we move on to incompatibility of Rh factor, that is erythroblastosis fetalis. Erythroblastosis fetalis is also known as Rh disease, or you can say the hemolytic disease of newborn. If you break up the term hemolytic, we get heme, that is blood, and lytic means destruction or lysis of RBC. So, this erythroblastosis fetalis is the disease which involves destruction of blood cells in fetus. Now let us see in detail how this disease occurs. So, Rh incapability has great significance in childbirth. If a woman is Rh negative and the man is Rh positive, if the man is Rh positive and the woman is Rh negative, the fetus may be Rh positive, having inherited the factor from his father. The Rh negative mother becomes sensitized by carrying Rh positive fetus within her body. In the fetal RBC, the red blood cells of the fetus is separated by placental barrier, embryonic chorion, from the mother's blood. Now, on the surface of the RBC, we have a protein molecule antigen called D antigen, making the fetal blood Rh positive. But on the other side of the placental barrier, we have the mother's blood lacking the D antigen on the RBC. So, the mother's RBC are devoid of D antigen on the surface of RBC. During parturition, that is childbirth, the placenta barrier breaks and creates the passage of blood to mingle. During mingling or mixing, the flow will be in both ways. We are more convinced that fetal blood entering into the mother circulation. So, fetal blood having D antigen on RBC will enter into the mother's blood circulation. The mother do not have D antigen on the surface of the RBC. So, the immune system of the mother will consider the D antigen as a foreign body and will launch the immune attack. This immune system response is Rh sensitizing. The mother's blood is sensitizing the Rh positive blood. The D antigen recognize the B cells and the B cells which transform into plasma B cells. This plasma cells secrete antibodies for D antigen. IgM is secreted first, and this is the initial response, and usually no effects are associated with exposure of the mother to Rh positive antigen during the first childbirth. So, if the mother gets pregnant, because IgG are produced after several hours, the initially produced IgM will not cause any side effect on the newborn. But after the delivery of the newborn, thousands of antibodies are produced by the D antigen. Subsequent Rh positive children carried by the same mother may be exposed to the antibodies produced by the mother against the Rh antigen. Here the antibody produced is IgG, which are carried across the placenta into the fetal circulation. This causes the hemolysis of fetal RBC, resulting in hemolytic jaundice and anemia. So this is called as erythroblastosis fetalis or hemolysis of the newborn. So how you can prevent this erythroblastosis fetalis? If the mother is Rh negative and the fetus is Rh positive, the anti-D antibody should be administered to the mother at 28th and 34th week of gestation as a prophylactic measure. If the Rh negative mother delivers Rh positive child, then anti-D antibody should be administered to the mother soon after delivery. This will develop a passive immunity and prevents the formation of anti-D antibodies in the mother's blood by destroying the Rh fetal RBC before the mother's immune system is sensitized. This has to be done whenever the woman attains pregnancy. So this is the way to control erythroblastosis fetalis. Next we move on to how the Y chromosomes determines the male development. Because male have XY chromosomes, so Y chromosome only determines the maleness. Current analysis of Y chromosome has revealed numerous genes and regions with potential genetic functions. Some genes with or without homologous counterparts are seen in X. Present at the both ends of the Y chromosomes, there are pseudo-autosomal region, that is PAR, that are similar to with the region of X chromosomes, which synapase, that means combines and recombine during meiosis. So during meiosis, this PARS region combine and recombine. The remaining 95% of the Y chromosome is referred to as non-combining region. That means the part which does not combine is referred as non-combining region of Y. It is denoted as NRY. The NRY is divided equally into, so this NRY genes are divided equally into functional genes and non-functional genes. Functional genes are euchromatic, true, and non-functional genes are heterochromatic. Within the euchromatic region is a gene, a special gene called sex-determining region Y, SRY, sex-determining region Y. In human, absence of Y chromosome inevitably leads to female development, and this SRY gene is absent in X chromosome. The gene product of SRY is the testis determining factor, TDF. So this SRY gene is produced from the testis, which represents the maleness. It is present in the adult male testis. So this SRY gene is absent in X chromosome, whereas it is present in Y chromosome. Next we move on to gene andromorph. What is gene andromorph? Gynoecium, andro, it's a fusion of male and female. These individuals have the parts of their body expressing male character as well as the other parts of the body expressing the female character. So you can see in these pictures, so male and female tissues are combined to form a single person. The organism is made up of tissues of male and female genotype and represents a different pattern. Next we move on to dosage compensation, or we can call it as bar body. So what is dosage compensation? What is dosage compensation? So we can able to see XX and XY chromosome. You see here there are 2X, here it is 1X. How it get compensated? So you can see this in detail. In 1949, Barr and Bertram first observed a condensed body in the nerve cells of the female cat, which was absent in the male. So 1949, Barr and Bertram found a condensed body in female cat which is absent in male. This condensed body was called as sex chromatin by them, and later it was called as Barr body. In XY chromosomal system of sex determination, males have only one X chromosome whereas the female has two. A question arises: how does the organism compensate for this dosage difference between the sexes? In mammals, the necessary dosage compensation is accomplished by the inactivation of one X chromosome in females. So the one X chromosome is inactivated in females, so that both males and females have only one functional, that means useful X chromosomes per cell. Mary Land suggested that bar bodies represented an inactive chromosome. These bar bodies are nothing but the inactive chromosome, which in females becomes tightly coiled into a heterochromatin, a condensed and visible form of chromatin that is called as Land hypothesis. The number of bar body observed in cell was one less than the number of X chromosomes. So X0 or XO females have no bar body because only one X is present, whereas XXY males have one bar body because they have two X chromosomes. So the number of bar bodies follows a rule, that is n-1 rule, where n is the total number of chromosomes present, X chromosomes present. Next, we move on to haplodiploidy condition in honeybees. So, in hymenopteran insects such as honeybees, ants, wasps, a mechanism of sex determination called haplodiploidy mechanism of sex determination is found. In this system, the sex of offerings is determined by number of sets of chromosomes it receives. So it lays two types of eggs. One is the fertilized egg, another one is the unfertilized eggs. Fertilized eggs develops into the females, that is the queen and worker, whereas unfertilized egg develops into male, that is drones by parthenogenesis. What is meant by parthenogenesis? Development of egg without the act of fertilization. It means that the males have half the number of chromosome haploid and the females have double the number, that is diploid. Hence the name haplodiploid. Haploid means 1N, diploid means 2N. This mode of sex determination facilitates the evolution of sociality in which only one diploid female becomes a queen and lays the eggs for the colony. So this determination, sex determination in honeybees helps to find out the queen. All the other females which are diploid having underdeveloped forms from the fertilized eggs helps to raise the queen's egg and so contribute to the queen's reproductive success and indirectly to their own. This phenomenon is known as kin selection. So from the fertilized eggs, females are produced, that means queen and worker. Out of that all the females, only one will be the queen by eating the royal jelly. So one will be the queen and others will be the workers. So workers can contribute their life to the queen's reproductive success, and this is called as kin selection. The queen constructs the social environment by releasing the hormone that suppresses the fertility of the workers. Next we move on to how the Y chromosomes determines the male development. Because male have XY chromosomes, so Y chromosome only determines the maleness. Current analysis of Y chromosome has revealed numerous genes and regions with potential genetic functions. Some genes with or without homologous counterparts are seen in X. Present at the both ends of the Y chromosomes, there are pseudo-autosomal region, that is PAR, that are similar to with the region of X chromosomes, which synapase, that means combines and recombine during meiosis. So during meiosis, this PARS region combine and recombine. The remaining 95% of the Y chromosome is referred to as non-combining region. That means the part which does not combine is referred as non-combining region of Y. It is denoted as NRY. The NRY is divided equally into, so this NRY genes are divided equally into functional genes and non-functional genes. Functional genes are euchromatic, true, and non-functional genes are heterochromatic. Within the euchromatic region is a special gene called sex determining region Y, SRY, sex determining region Y. In human, absence of Y chromosome inevitably leads to female development, and this SRY gene is absent in X chromosome. The gene product of SRY is the testis determining factor, TDF. So this SRY gene is produced from the testis, which represents the maleness. It is present in the adult male testis. So this SRY gene is absent in X chromosome, whereas it is present in Y chromosome. Next we move on to gene andromorph. What is gene andromorph? Gynoecium, andro, it's a fusion of male and female. These individuals have the parts of their body expressing male character as well as the other parts of the body expressing the female character. So you can see in these pictures, so male and female tissues are combined to form a single person. The organism is made up of tissues of male and female genotype and represents a different pattern. Next we move on to dosage compensation, or we can call it as bar body. So what is dosage compensation? What is dosage compensation? So we can able to see XX and XY chromosome. Is it here there are two X, here it is one X. How it get compensated? So you can see this in detail. In 1949, Barr and Bertram first observed a condensed body in the nerve cells of the female cat, which was absent in the male. So, 1949, Barr and Bertram found a condensed body in female cat, which is absent in male. This condensed body was called as sex chromatin by them, and later it was called as Barr body. In XY chromosomal system of sex determination, males have only one X chromosome, whereas the female has two. A question arises: how does the organism compensate for this dosage difference between the sexes? In mammals, the necessary dosage compensation is accomplished by the inactivation of one X chromosome in females. So the one X chromosome is inactivated in females, so that both males and females have only one functional, that means useful X chromosomes per cell. Mary Land suggested that Barr bodies represented an inactive chromosome. These Barr bodies are nothing but the inactive chromosome, which in females becomes tightly coiled into a heterochromatin, a condensed and visible form of chromatin. It is called as Land hypothesis. The number of Barr body observed in cell was one less than the number of X chromosomes. So X0 or XO females have no Barr body because only one X is present, whereas XXY males have one Barr body because they have two X chromosomes. So the number of Barr bodies follows a rule, that is n minus 1 rule, where n is the total number of chromosomes present, X chromosomes present. Next we move on to haplodiploidy condition in honeybees. So in hymenopteran insects such as honeybees, ants, wasps, a mechanism of sex determination called haplodiploidy mechanism of sex determination is found. In this system, the sex of offerings is determined by number of sets of chromosomes it receives. So it lays two types of eggs. One is the fertilized egg, another one is the unfertilized eggs. Fertilized eggs develops into the females, that is the queen and worker, whereas unfertilized egg develops into male, that is drones by parthenogenesis. What is meant by parthenogenesis? Development of egg without the act of fertilization. It means that the males have half the number of chromosome haploid and the females have double the number, that is diploid. Hence the name haplodiploid. Haploid means 1n, diploid means 2n. This mode of sex determination facilitates the evolution of sociality in which only one diploid female becomes a queen and lays the eggs for the colony. So this determination, sex determination in honeybees helps to find out the queen. All the other females which are diploid having underdeveloped forms from the fertilized eggs helps to raise the queen's egg and so contribute to the queen's reproductive success and indirectly to their own. This phenomenon is known as kin selection. So from the fertilized eggs, females are produced, that means queen and worker. Out of that all the females, only one will be the queen by eating the royal jelly. So one will be the queen and others will be the workers. So workers can contribute their life to the queen's reproductive success, and this is called as kin selection. The queen constructs the social environment by releasing the hormone that suppresses the fertility of the workers. Sex-linked inheritance. The inheritance of a trait that is determined by a gene located on one of the sex chromosomes is called sex-linked inheritance. The genes present on the differential region of X or Y chromosome are called sex-linked genes. The genes present in the differential region of X chromosomes are X-linked genes. The X-linked genes have no corresponding alleles in the Y chromosome. The genes present in the differential region of Y chromosome are called Y-linked genes or Hollandric genes. The Y-linked genes have no corresponding allele in X chromosome. The Y-linked genes inherit along with Y chromosome and they phenotypically express only in the male sex. Sex-linked inherited traits are more common in males than in females because Male are hemizygous. Hemi means half. They have one X chromosome, another Y chromosome, and therefore express the trait. Trait means character. When they inherit one mutant allele. The X-linked and Y-linked gene in the differential region, that is a non-homologous region of a chromosome, do not undergo pairing or crossing over during meiosis. So the inheritance of X or Y-linked genes is called sex-linked inheritance because X and Y are sex-linked chromosomes. So the inheritance of X and Y-linked genes are called sex-linked inheritance. Inheritance of X-linked genes. So first we see inheritance of X-linked genes. So for this X-linked genes, the best example is hemophilia and color blindness. First we see hemophilia. Hemophilia is commonly known as bleeder's disease, which is more common in men than women. The hereditary disease was first observed or reported by John Cotto in 1803. Hemophilia is caused by a recessive X-linked gene. There are two types, recessive and dominant. So hemophilia is a recessive X-linked gene. A person with a recessive gene for hemophilia lacks a normal clotting substance. So normally our blood is having a blood clotting substances that is called as thromboplastin in blood. Hence, minor injuries cause continuous bleeding, leading to death. The females are the carriers of the disease and you transmit the disease to 50% of their sons, even if the male parent is normal. So here, the females are the carriers. They carry this disease to their sons. Hemophilia follows the characteristics crisscross pattern of inheritance. Next, we move on to color blindness. Color blindness in man or human beings. Here, color blindness is a sex-linked recessive disease. Already I told you there are two characters, dominant and recessive. Color blindness is a sex-linked recessive disease. This gene is located on the X chromosomes. So color blindness, it is located in X chromosomes. In females, there are two X chromosomes, and in males, there are one X chromosomes. So the homozygous recessive females XC, XC, and hemizygous recessive males XC and Y. So affected person cannot able to distinguish red and green colors. In human beings, a dominant X-linked gene is necessary for the formation of colors. So they can't able to distinguish red and green color. So it is more common in males than in females because the males have only one X chromosome, X and Y. So presence of even one allele will cause color blindness. So in color blindness, it is represented as XC. So if only one allele is present in males, then they will be the color blinded male and only one Y chromosome. But in females, they have two X chromosomes. So female will be color blind only if both X have alleles for color blindness. That means XC and XC. But in this case, you see, only one X is having color blindness and another X is normal. In this case, the female will be a carrier. So if a female has only one allele, the female will be a carrier. She will transmit the disease from one generation to the another generation. Next, we move on to the different types of inheritance of color blindness. The first one is the marriage between colorblind man and normal vision women. So normal female has two X gametes, whereas colorblind male has one XC and one Y gamete, because XC having colorblind allele. So if you see the offerings in a Punnett square, we will put a Punnett square, and here is the males having XC and Y chromosomes, and in female 2X chromosome. If you cross these two, male and female, it will produce XC and X. It is a carrier female because only one X is having color blindness. Next is XY, that is the normal male. Next is XC and X. It is also the carrier female. Next is X and Y. It is normal male. If you see these two carriers, so the result is two female carrier and two normal males are produced at the end of F1 generation. Second case is a marriage between a normal visioned man and a colorblind woman. So colorblind woman is having XC parents and the gametes produced are XC and XC. Because they are colorblind, both X's is having the C, that is a colorblind alleles. The normal vision man is having X and Y chromosomes. Normal vision, there is no color blindness in X. Okay. So next is if you see the offerings, here is the male X and Y, here is the female, both XC and XC. If you cross these two, XC and X, so that is the carrier female. And next is the XC and X, it is also the carrier female because only one X is having the colorblind allele. Here XC and Y, it is a colorblind male. Next is also XC and Y, it is a colorblind male because the X chromosome is having the colorblind. These two, XC and X, are female carriers. Two female carriers and two colorblind males are produced at the end of F1 generation in this case. Next is the third case. This is male. The male is a normal man and a female is the carrier woman. This we might see in the F1 generation of the first case. So the parents are XC and X and the normal male is X and Y. If you see the offerings, the male has X and Y and the female has XC and X. So the first one is the XC and X, that is the carrier, and here XC and Y, that is the colorblind man. X and X, it is a normal female, and X and Y, it is a normal male. Here the result produced at the F2 generation is one carrier girl and one normal girl, one colorblind boy and one normal boy. Next, we move on to the fourth case, that is the F2 generation of the second one, the carrier mother and the colorblind father. Here the parents are XC and X because she is a carrier, so only one X is having that colorblind allele and another X is the normal, and colorblind father and XC is having only one X, this X is having C and Y normal chromosome. So the progeny produced or here is the male XC and Y and female is the XC and X. The first one is the XC and XC. So it is a colorblind female. Next one is XC and X. It is a carrier female. XC and Y. It is a colorblind male. And next is the X and Y. It is a normal female. So at the end of F2 generation, so this will produce one colorblind daughter or female and one carrier female or daughter and one colorblind male or one normal male. Next, we move on to inheritance of Y-linked genes. Genes in the non-homologous region of the Y chromosome are inherited directly from male to male. In human, the Y-linked or Ollandric gene for hypertrichosis. Hypertrichosis means excessive development of hairs on pinna of the ear. So in this picture, you can able to see the excessive development of hair in the pinna of this man. So these are transmitted directly from the father to the son, because males inherit the Y chromosome, because male is only having the Y chromosome. Female has 2 X chromosome, whereas the male has 1 X and Y chromosome. So maleness is characterized only by this Y chromosome from the father. So females inherits only X chromosomes from the father, and they are not affected. Next, we want to karyotyping. What is karyotyping? Karyotyping is a technique through which a complete set of chromosomes is separated from a cell, and the chromosomes are arranged in pairs. An ideogram refers to the diagrammatic representation of a chromosome. The diagrammatic representation of chromosome is called as ideogram. So how we can prepare this karyotype? Geo and Levin in the year 1960 described a simple method of culturing these lymphocytes. A lymphocyte is a type of WBC from the human blood. Mitosis is induced, followed by the addition of colchicine to arrest the cell division at metaphase stage. So there are different stages in mitosis: prophase, metaphase, anaphase, and telophase. So in metaphase stage, the cell division is arrested, and the suitable spread of metaphase chromosomes is photographed. So the individual chromosomes are cut from the photograph and are arranged in an orderly fashion in homologous pairs. This arrangement is called karyotype. Chromosome banding permits structural definitions and differentiation of chromosomes. So what is the use of this karyotyping? Applications of karyotyping involves: number 1 is it helps in gender identification. It is used to detect the chromosome aberration, such as, that means chromosomal abnormalities such as deletion, if a part of chromosome is deleted, duplication, translocation, non-disjunction of chromosomes. It also helps to identify the abnormalities of chromosomes like aneuploidy. What is aneuploidy? Loss or gain of one chromosome. It is also used in the predicting the evolutionary relationship between the species. Karyotypes helps in predicting the evolutionary relationship between the species. And last is the genetic diseases in human beings can be detected by this karyotyping. So I repeat the applications. First one is the gender identification. Second one, to detect the chromosomal aberration. Third one, to find out the abnormalities in chromosome like aneuploidy. Fourth one is the predicting the evolutionary relationship. And fifth one, to find out the genetic diseases in human beings. So how can we do this human karyotype? Depending upon the position of the centromere. Centromere, what is centromere? The centromere is present in between the chromosome. There are 2 chromatids, and the center there is an connecting part. That connecting part is called centromere. So depending on the position of the centromere and the relative length of 2 arms, human chromosomes are of 3 types: metacentric, submetacentric, and acrocentric. Metacentric means centromere is at the center and the 2 arms are equal. Submetacentric means one arm is long and another arm is short. And acrocentric means one arm is too short and another arm is too long. So another method is also there, that is called a stelocentric, where only one arm is present. The centromere is present at the tip. This you might have studied in your lower classes. So the photograph of chromosomes are arranged in the order of the descending length in different groups. So human karyotype, the chromosomes are arranged in different groups. So A to G. So the total number of chromosomes is 23 pairs. Okay. So these 23 pairs, they are arranged in A to G groups. 1, 2, 3. The first 3 are in A group. 4, 5 is in B group. C group is the very largest group. It consists of 6, 7, 8, 9, 10, 11 and 12. And this C group also has the sex chromosome X. 13, 14 and 15 belongs to D group. 16, 17, 18 belongs to E group. 19 and 20 F group. 21 and 22 G group. Here in G group, the Y chromosome, sex chromosomes is present. So out of 23 pairs, 22 pairs are autosomes and 1 pair is sex chromosomes. So 22 pairs are arranged in different groups from A to G, and the sex chromosome X and Y, X is present at the C group whereas Y is present in the G group. So question will be asked like this. So where this X and Y chromosomes are present in which group? So in which group the X and Y chromosomes are present? So you should answer C and G. Next we move on to pedigree analysis. Pedigree is a family tree drawn with standard genetic symbols showing the inheritance pathway for specific phenotype characters. Pedigree analysis is a study of traits as they have appeared in the family line of several past generations. So with the help of this pedigree analysis, we can able to find out our super seniors. That means the family trees can be identified. So we can able to find out நம்ம வம்சத்துல எல்லாத்தையும் ஃபுல்லா எல்லாத்தையும் தெரிஞ்சுக்கலாம். Okay, so this pedigree analysis can be given by different symbols which can be used in the pedigree charts. So the symbols, so in this picture you can able to see different symbols. Male on square, female it's a round. So mating, male and female, this you can able to read out from your, this table is given in your book. So book link is sent to you, so you may read this. Okay, next we move on to genetic disorders. Next we see genetic disorders. A genetic disorder is a disease or syndrome that is caused by the abnormality of an individual DNA. Genetic disorders are of two types, namely Mendelian disorder and chromosomal disorders. Mendelian disorders includes hemophilia, sickle cell anemia, color blindness, phenylketonuria, thalassemia, and cystic fibrosis. Chromosomal disorders includes Down syndrome, Klippel-Trenaunay syndrome, Turner syndrome, and Patterson syndrome. We will see these disorders one by one. First, we see the Mendelian disorders. What is Mendelian disorder? Alteration or mutation in a single gene causes Mendelian disorders. These disorders are transmitted to the offsprings on the same line as the Mendelian pattern of inheritance. Some of the examples are hemophilia, color blindness, sickle cell anemia, and thalassemia. The pattern of inheritance of Mendelian disorders can be traced in a family by pedigree analysis. That means by family tree. This Mendelian disorders may be dominant or it may be recessive. Pedigree analysis helps to understand whether the trait is dominant or recessive. Next we see phenylketonuria. It is an inborn error of phenylalanine metabolism caused due to a pair of autosomal recessive genes. It is caused due to mutation in the gene PAH, that is phenylalanine hydroxylase gene, located on chromosome 12 for the hepatic enzyme phenylalanine hydroxylase. This enzyme is essential for conversion of phenylalanine to tyrosine in a normal person. But in affected individual, this enzyme lacks. As a result, phenylalanine accumulates and converts this into phenylpyruvic acid and other derivatives. So, in phenylketonuria patient, this phenylalanine is converted into phenylpyruvic acid. If you see the characteristics of the phenylketonuria patients, they are have severe mental retardation, light pigmentation of skin and color. Phenylpyruvic acid and other derivatives accumulates in brain, resulting in mental retardation. These are also excreted through urine because of poor absorption of kidneys. So this phenylpyruvic acid is excreted through urine. Next we see thalassemia. Thalassemia is an autosomal recessive disorder. It is caused by gene mutation resulting in excessive destruction of RBCs due to the formation of abnormal hemoglobin molecules. Normally, the hemoglobin is composed of 4 polypeptide chains, 2 alpha and 2 beta globulin chains. Thalassemia patients have defect in either the alpha or beta globulin chain, causing the production of abnormal hemoglobin molecule resulting in anemia. So thalassemia is caused due to destruction of RBC. This thalassemia is classified into alpha and beta based on which chain of hemoglobin molecule is affected. First one is the alpha thalassemia. Here, production of alpha globulin chain is affected. In alpha thalassemia, alpha globulin chain is affected and is controlled by two closely linked genes HbA1 and HbA2 on chromosome 16. Mutation or deletion of one or more of the 4 genes causes this disease. Next one is the beta thalassemia. In beta thalassemia, we can see here in this picture the beta thalassemia. In beta thalassemia, the production of beta globulin chain is affected. It is controlled by a single gene HBB, hemoglobin B on chromosome 11 of each parent. Mutation of one or both the genes causes this disease. The other Mendelian disorder is albinism. Albinism is an inborn error of metabolism caused due to autosomal recessive gene. Melanin pigment is responsible for skin color. Absence of melanin results in a condition called albinism. A person with the recessive alleles lacks the tyrosinase enzyme system, which is required for conversion of dihydroxyphenylalanine, that is DOPA, into melanin pigment inside the melanocytes. Albino means albinism. The melanocytes are present in normal numbers in their skin, hair, iris, etc. Iris is a part of a eye, but lack melanin pigment. So you might see this equation 3,4-dihydroxyphenylalanine, that is DOPA, is converted into melanin by the enzyme tyrosinase. Tyrosinase is responsible for production of melanin in our skin. Next is the Huntington chorea. It is inherited as an autosomal dominant lethal gene in man. It is characterized by involuntary jerking of the body and progressive degeneration of nervous system, accompanied by gradual mental and physical deterioration. That is, physically and mentally the person is affected. The patient with this disease usually die between the age of 35 and 40. These are some of the Mendelian disorders. Next is the chromosomal abnormalities. Each human diploid, that is a 2N body cell, has 46 chromosomes, that is 23 pairs. Chromosomal disorders are caused by errors in the number or the structure of chromosome. Chromosomal anomalies usually occur when there is an error in cell division. Failure of chromatid to segregate during cell division, resulting in gain or loss of one or more chromosome, is called aneuploidy. Aneuploidy means addition or loss of one or more chromosome, because The chromatids fail to segregate during cell division. So it is caused by non-disjunction of chromosomes. Group of signs and symptoms that occur together and characterize a particular abnormality is called syndrome. So what is called syndrome? It is a group of signs and symptoms that characterize a particular abnormality is called as syndrome. So in human, Down syndrome, Turner syndrome, Klippel-Trenaunay syndrome, Patus syndrome, these are some of the examples of chromosomal disorders. This chromosomal disorder is divided into two, autosomal and allosomal. Autosomal means it have changes in their body cells, and allosomal means meiotic disjunction of sex chromosomes takes place. So autosomal aneuploidy includes Patus syndrome, Down syndrome, and allosomal abnormalities includes Klippel-Trenaunay syndrome and Turner syndrome. We will see one by one. First is the Patus syndrome. Trisomic condition of chromosome 13 results in Patus syndrome. The non-meiotic disjunction is thought to be the cause for this chromosomal abnormality. It is characterized by multiple and severe body malformation as well as profound mental deficiency. This syndrome causes small head with small eyes, cleft palate. Cleft means divided. Palate means present inside the mouth. Malformation of the brain and the internal organs are some of the symptoms of this disease. The next chromosomal abnormality is the Down syndrome. It is also called as trisomy 21. Trisomic condition of chromosome 21 results in Down syndrome. The genetic constitution of this have 45A plus XX, or it may be XY. Totally it is 47 chromosomes. So the characteristic features of this Down syndrome is severe mental retardation, defective development of central nervous system, increased separation between the eyes, flattened nose, ears are malformed. Mouth is constantly open and the tongue protrudes. And there are some congenital heart diseases also occur in this Down syndrome. You can see this in the pictures, how the child will be. The next chromosomal disorder is the Klippel-Trenaunay syndrome. This genetic disorder is due to the presence of one additional copy of X chromosome, resulting in a karyotype of 47XXY. Normally that is 46 chromosome. Here there is 47, one extra X chromosome in male. These persons have 47 chromosomes. They are usually sterile males, tall, obese, with long limbs, high-pitched voice, and developed genitalia, and have feeble breast development. You can see this in the picture. That is one extra chromosome, X chromosome. Next is the Turner syndrome. This genetic disorder is due to loss of one X chromosome, resulting in a karyotype of 45X. Loss of one chromosome, 46 minus 1, that is 45 X chromosomes. It is of females. Persons with this syndrome have sterile females, low stature, webbed neck, and developed breast, rudimentary gonads, lack of menstrual cycle during puberty. These are the main symptoms of this syndrome. You can see this in the pictures. Students, I hope you understood the lesson. So that's all. We finished the 4th chapter, and please go through that. Don't waste your time. Keep on enriching your studies. Thank you.